Thank you, everyone, and let me add my congratulations and thanks to the organizers. Anti-VEGF biologics for the treatment of neovascular AMD have been with us for 11 years and 7 months as of now, which is approximately 139 injections in retina time. And it's not surprising that together with the significant numbers of efforts being made to improve treatment and add additional treatments and alternative targets, the other part of the market that's developing is the market that is around chronic management of these patients, because the demographics would suggest that the patients that had wet AMD 11 years and 7 months ago, 50% of them are still with us. And as you think about progressing these treatments, into younger patient populations like diabetes, where there may be 3 decades of treatment, alternative ways of administering anti-VEGF, which is foundational to the treatment of wet AMD, are going to need to be available. Let's think about any other area where we have serious disease that is also that works alongside with patient-administered treatments that allow for a long-standing treatment for the treatment of wet AMD. So we have been working for a few years now on developing a topical eye drop that targets VEGF, and we were fortunate enough to identify PAN-90806, a small molecule antagonist to VEGF with an unusual group of characteristics for topical eye delivery. Our PK has shown that it has a peripheral route of administration through the choroidal circulation, produces excellent target tissue concentrations at hundreds to thousands-fold the KD of the molecule at the central retina and central choroid. And that concentrations are actually sustained out 17 hours past dose, which is the longest period that we've measured, allowing us to consider once or twice a day treatment. And it also has very low systemic exposure when measurable, and it performs as well as an IVT antibody in animal models of both ischemic as well as choroidal neovascularization. We have filed an IND, and we have, in 2014, initiated treatment in a phase 1/2 dose-ranging study. This is in treatment-naive patients with neovascular AMD. There are 2 stages to this study. The first dose ranging in 40 patients over 5 doses, monotherapy up to 8 weeks with Lucentis rescue available. We have recently completed enrollment and last patient out in that group, and we will give you at least, even though the trial is ongoing, a little bit of hint of what we've seen so far. Stage 2 is 10 patients where each patient gets a baseline intravitreal injection with Lucentis, followed a week later by up to 3 months of treatment with an eye drop, again with Lucentis rescue available. To date, we have 6 patients in that 10-patient cohort. 4 patients have completed, and to date, there have been no intravitreal rescues. We expect all of this data to be available to us in the first quarter or second quarter of next year. So we have reviewed the cases together with some of our retina experts, and they have confirmed that we have biological activity in these patients over 8 weeks across all of the dose arms, from lowest to highest. There have been no treatment-related systemic AEs to date. Maximally tolerated dose has been identified with the current formulation at the 8-week time point. The AEs are limited to reversible corneal changes, really visible by corneal fluorescein staining, punctate erosions in some of the patients at the higher dose frequency arms. The safety is undergoing continuing assessment by the Safety Monitoring Committee today, and we expect full results in the first or second quarter of next year, as I mentioned earlier. I'm going to give you maybe some permission to believe, and 2 cases does not make proof, but it does give you a sense of possibility and potential. So this is a patient treated at 2 mg per ml once a day that had a occult lesion with moderate thickening and some vision loss at baseline. To orient you, the top row are the images from baseline, week 1, week 2, week 4, and then the final visit on therapy at week 8. We follow up at 1 week and 1 month post-therapy as well. What you see is that at week 8, there's about a 70 micron thinning and resolution of the subretinal fluid, And actually, this patient was not treated for the month following and is stable and has continuing good vision. This is the second patient, 1188. This one has a significant amount of intraretinal fluid. You see day 1 and week 8. At week 8, there's 140 micron thinning. This is eye drops only. And you see reaccumulation of fluid at 1 week and 1 month after cessation of the treatment. Now, obviously, this is not proof, as I said, and we'll have the full data set soon, but it does give us confidence in moving forward with development, not only for neovascular AMD, but for diabetic retinopathy and other VEGF-dependent eye diseases. We are also currently enrolling in a phase 1 dose-ranging study at the maximally tolerated dose here of the eye drop in patients with PDR with and without DME. The goal here is to show whether or not adequate therapeutic concentrations are reaching the retina so that we can see evidence of either neovascular regression or improvement of edema. Now, the other thing that's been going on is we've learned that we're a formulation and drug delivery house as well, and we've done a significant amount of work on identifying an optimized formulation that could allow us to move into phase 2 trials with a better formulation and a lower potential for corneal adverse events. And I'm pleased to report that we've just recently finished a non-human human primate study that shows a 3/4 to a log and a half reduction in corneal exposure that is not dose dependent. So at doses from 1 to 10 mg per mil, we're getting uniformly low exposures across the cornea, and at the same time has enhanced our ability to deliver drug to the back of the eye, in this case at 2 to 10 times the dose in the primate that we had been able to deliver previously across the dose range. So in summary, what we'll be doing is really moving back to the clinic in 2016-17 with an optimized formulation and the ability to confirm tolerability and safety in monotherapy. It's the best way to see your dose response and the safety and efficacy of your drug because it has to do something on its own. And then demonstrate safety in a phase 2b trial that's adequately powered in a maintenance setting where these drugs could exist alongside of physician-administered drugs in the chronic management of neovascular eye diseases. I'd like to particularly thank my board and my investors, SV Life Sciences, who's been there with us from the beginning, Third Rock Ventures and Novo Ventures that joined us last year in our Series B financing. And these are the people that make it happen, and they're all here, and I'm eternally grateful to you for being who you are. Thank you very much.